As we all know, climate change has grown interest during these past centuries. It has become one of the biggest challenges for governments and managers in the last few years, as it may significantly accelerate biodiversity loss. Due to anthropogenic activities, our oceans are currently experiencing severe changes and a huge increase in atmospheric greenhouse gases concentration, particularly carbon dioxide, causing an increase in water temperature and acidity in the oceans. Responses to ocean warming and acidification may vary across the life history of organisms, embryonic and juvenile stages being the most vulnerable. Elasmobranchs are one of the most ecologically important species as they fill many niches across a wide range of biota. They typically occupy high trophic levels as top or mesopredators, thus playing a key role in the health of marine ecosystems. These species are characterized by a slow growth, relatively long lifespans, and tend to have higher survival rates during the juvenile stages when compared to telespecies. Even if their relatively low generational turnover rate allowed them to evolve during all these millennia, surviving 5 major mass extinction events, it makes them particularly vulnerable to today's rapid changes in the environment. Skates and rays have short, stiff head trunk regions, forming a disc with slender tail and therefore swim using the pectoral fins in undulatory or oscillatory motions. Locomotion is a key process in nearly all fishes, including elasmobranchs, that is involved in vital functions such as reproduction, migration, predator avoidance, run ventilation, and small-scale movement. Two major locomotoral behaviors are likely to increase survival rates in elasmobranchs: steady swimming and burst performance. During steady swimming, fishes may modulate their speed and can often optimize energetic costs for large-scale movements by selecting an optimal velocity. On the other hand, fishes may undertake energetically costly movements such as fast starts and bursts to escape a predator or to capture a prey. It wasn't until recently that experimental data showed that high levels of carbon dioxide can affect elasmobranch fishes directly by causing morphological, behavioral, and physiological anomalies, showing that warming had a negative effect on the mineralization of the pectoral fins as the density of apatite decreased. One of the possible causes of a reduction in mineralization in the pectoral fins of skates may be attributed to a mismatch between high growth in juveniles at higher temperatures and mineralization processes that might proceed at low rates. A decrease in mineralization of the cartilage in the pectoral fins produces higher flexibility during movement, but the fish needs to actively stiffen the fins to achieve higher swimming speeds, thus increasing metabolic costs during swimming. Smaller body size is known as one of the universal responses to climate change, as well as a reduced aerobic scope for activity and increased stress. It has been suggested that some fish species that live close to their thermal optimum may experience a reduction in the amount of available energy for aerobic activities, such as locomotion, because of ocean warming. Elasmobranch species are fast swimmers and obligate run ventilators, having the highest measures of metabolic rates to maintain heterothermy, and therefore are expected to have a limited amount of energy to cope with environmental challenges than less active species. High levels of carbon dioxide increases mineralization of the crura, modified pelvic fins that some rays use to walk underwater, and jaws in little skates. Skates might even gain an advantage as high mineralization of the crura is a good predictor of the ability of batoids to walk underwater. But at the same time, the weight of a denser skeleton reduces buoyancy, which increases energetic cost of swimming. Since elasmobranchs do not have a swim bladder, they need to swim in order to create hydrodynamic lift, or even hover in the water column. Therefore, a heavier skeleton is expected to increase cost of locomotion and turning speed. Studies have not reported any body malformation linked to acidification in elasmobranchs yet, and it has been suggested that the elasmobranch embryos might be protected from acidification during the earliest stage of development, while inside their mothers or in a closed egg case. Experiments test the combined effects of ocean warming and acidification on locomotor performance in skates, showing a prolonged recovery time from intense exercise and higher energetic costs to escape from predators. All of this is making skates vulnerable to predators, forcing them to explore the environment to seek additional food. This combination also reduces elasmobranch's ability to use olfactory cues, increasing time to find food and therefore swimming activities. As our planet's climate continues to evolve, these incredible creatures have demonstrated remarkable adaptations to survive in their changing environment. Marine organisms vary their physiological and morphological characteristics and, as a result, their individual response to ocean warming and ocean acidification differs significantly. Changes in locomotor performance, behavior, and geographical range at a large and small scale may have cascading and unexpected effects on biotic interaction, community structures, and ecosystem functioning. Current rates of change in temperature and pH are much faster than those experienced during previous climate shifts, especially of warming and acidification. These two, combined with other stressors such as habitat destruction, variability, and exploitation, may render the management and protection of this species difficult. To be able to forecast future outcomes and strategize on different measures to implement to manage whale populations, it is most important to identify key traits that impact swimming performance under climate change scenarios. Locomotor efficiency is a major trait determining the capacity of elasmobranchs to fare well in climate change. Even though ocean warming and acidification are considered to be some of the biggest threats to marine biodiversity, the individual and synergistic effects of these stressors on elasmobranch species like sharks and rays are still clearly understudied. These morphological and physiological impairments, in combination with hypoxia, pollution, fishing, and habitat destruction, may act to reduce elasmobranch survival.